Bismillahirrahmanirrahim. Today we will discuss T lymphocytes. In this lecture we will discuss its origin, from where they are originated, how they are developed, its early development, where they develop, and migration to the thymus, in maturation in thymus, and then deposition in lymph node and spleen. So original early development and migration to the thymus. All T cells originate from the haematopoietic stem cell HSC, which reside in the bone marrow. In some cases, the origin might be the fetal liver during embryonic development. These cells are also produced during fetal life from the liver, but in adult they are produced by the bone marrow. Haematopoietic stem cell then differentiate into multipotential progenitor MPP, which retain the potential to become both myeloid and lymphoid cells. T cell maturation in the thymus. Immature thymocytes go to the thymus to develop into fully mature T cell. Not all the thymus cells which are migrated from the bone marrow to the thymus, they all are not mature, but because some of them are die. We will discuss the reason why some become mature and some are become die. So some are destroyed by apoptosis during the development and maturation process. Here this is the multipotential haematopoietic stem cell. First, there is production of hemocytoblast. This cell is present in the bone marrow and in the stem cell. This cell has the capability to produce common myeloid progenitor, which is responsible for the production of RBC and other granulocytes and platelets. On the other hand, it is differentiated into common lymphoid progenitor, which either produce natural killer cell or small lymphocytes. These lymphocytes are then produce T cell or B cell. But today our discussion is limited to the T lymphocyte. We'll discuss the T lymphocyte when they are produced in the bone marrow, how they are shifted to the spleen and become mature T lymphocyte. As we already discussed, these cells are present in the bone marrow, are produced or originated in the bone marrow from the stem cell, haematopoietic stem cell. Then this haematopoietic stem cell is capable of producing myeloid stem cell and lymphoid stem cell. The haematopoietic stem cell has the capability to produce these two types of cells, either myeloid or lymphoid stem cell, and they also make its own copies. They don't become totally burn its capacity, but they also produce its copies for the upcoming situation. Now, as we are concerned with this lecture, in this lecture we will discuss T cells. So when these T cells are produced, they are added to the circulation and then they are transported to the thymus. There are factors which attract these cells towards the thymus, and these factors are thymopoietin and pituitary hormone, which is thymosin and thymolene, help make specialized type of T cell. These originally generated T lymphocyte in the bone marrow. They are attracted by these hormones and chemicals to the thymus and then develop or mature these cells to specialized type of T lymphocytes. When these T lymphocytes arrive in the thymus, they are immature. They neither have T cell receptor or no CD4 or CD molecule. That's why they are called double negative cell and they are immature. And maturation takes place in the thymus. This maturation is caused by the hormone which are produced by the thymus cell. These hormones are thymosin alpha, thymosin beta 4, somatomedin C, and thymopoietin. So these four hormones cause maturation of these immature cells, and these hormones are produced by the thymus cell. These hormones actually act on the nucleus of the T lymphocyte. As we know that the T lymphocyte is nucleated. So these hormones act on these four hormones act on the nucleus of the T lymphocyte. And due to this action of these hormones on the nucleus, as nucleus contain the DNA contain chromosome. So from the nucleus, the RAG1 and RAG2 are produced. This RAG1 and RAG2 then act on the chromosome of the nucleus on the chromosome which are present in the nucleus of these T lymphocytes. So first, these hormones act on the nucleus, produce RAG1, RAG2, and RAG1, RAG2 act on the chromosome and produce protein. These proteins are the RGA1 and RGA2. They are actually protein and they are called recombinant proteins. Their protein actually separate, shuffle, and rejoin the VDJ genes. This shuffling takes place inside B cell and T cells. So actually, this RAG1 and RAG2 are recombinant protein hormones, and they actually cause reshuffling of the protein which are present in the chromosome, and they are reshuffled. This reshuffling causes actually production of receptor and molecule on the surface of the T lymphocyte. This is a receptor which is caused by when reshuffling in the molecule occur and RGA and RGA2 act on these cells, they produce a receptor on the surface of T lymphocyte. This receptor has a portion which can attach to any antigen, and it can change its shape according to the shape of antigen. And also here on the surface of this immature T cell, under the action of this protein, a molecule CD8 or CD4 are produced. So when these two receptor and molecules are produced on one cell, the receptor or CD8 molecule is produced, or T cell receptor or CD4 is produced. Then it is expressed to the thymic cell. These cells which generate receptor and molecule on its surface, then they are cross-checked with the thymus cells. The thymus cell have receptor on their surface. These receptors are either MHC1 receptor or MHC2 receptor. So when these receptors are presented to the thymus cell, they check these receptor and molecules. If the molecule synthesis is correct and there is no derangement in the development process of these receptors on the T lymphocyte, they are marked OK or they are clear. So the MHC1 molecule, this is the MHC1 molecule, it is presented to the T lymphocyte which has receptor for the MHC1 molecule. On the other hand, this T cell is presented to another thymic cell which has receptor for the MHC2 major histocompatibility 2 complex, and the thymus cell check this molecule which is CD4, and if this is produced correctly and there is no abnormality developed during the stages of maturation, then they are accepted. So this mechanism, the identification and clearance of the cell and its molecule by the T lymphocyte, or coding of these cells by the T lymphocyte is called positive selection. This positive selection causes these cells not to reactor the MHC1 or MHC2 molecule which are present on every nucleated cell in the body, which we have discussed in the major histocompatibility lectures 1 and 2. Now these cells also contain receptors. These receptors are also checked by the thymus cell, and their acceptance or rejection is checked. If the receptor is developed properly and it is matching with the body tissue, they are cleared or coded, and they are declared compatible with the immune system of the body, and this mechanism is also called positive regulation. But selection, and if they are not developed properly, the receptor or either the molecule on any cell, when they are not developed properly, they are rejected, and this is called negative selection. When they are rejected, they pass through apoptosis or cellular death. This cell which doesn't match the body immune system which is present outside the body, then they are rejected and these cells become die. And this is called apoptosis or cellular death. Another event which happened here in the thymus, when the CD4 molecule attached to the thymus receptor, and when it mark it OK, so then on this cell only CD4 molecule, this cell then produce CD4 molecule, and the CD8 are inhibited. So this inhibition of CD8 molecule is called negative regulation. In the same case, if CD8 is accepted and CD4 is rejected, so this is called negative or down regulation for the CD8. Down regulation means there is reduction or totally prevention of development of these molecules. Now when these molecules, these cells are mature in the thymus, they are then added from thymus to the circulation, and through circulation they are either deposited in the cortex of lymph node or in the white pulp of the spleen, where they then perform its immune response, or when it is activated by the antigen in the body. So either the T or the B cell then attack on these antigen, which are expressed on the either on the MHC1 molecule or MHC2 molecule, which we have discussed in the previous lecture. So thank you. Please subscribe the channel and press the bell icon for upcoming notification, and leave your valuable comments for improvement of the channel. Thank you once again.